Hey guys, welcome to Biology from Home. For this lecture, I'm going to be going over notes in your new packet, Unit 11. We're going to talk about Part 1. So I think I'm going to go through bacteria, the bacteria part. It goes to page like 2 in the very top. And then the next video, I'm going to do viruses. And then we'll have probably a homework assignment to recap all that before moving on to part 2, protists and fungi. Okay, so I'm just going to pull up the PowerPoint and go through it so you guys can follow along and start filling in your notes for that section. So give me 1 second. Okay, and let me move that. There we go. Okay. So I'll just move my face over here. Okay. Okay, so this is on bacteria. These are early kingdoms. I will post this video up on your Google Classroom so that we don't waste time on this. So bacteria, the good, bad, the ugly. Ooh. All right, so there's 3 different kind of shapes for bacteria. The first shape is cocci, like Streptococcus. So these are spheres. Bacilli are rounds and Spirilla are spirals. In terms of cell walls, they can be either gram-positive or gram-negative. And in terms of movement, they can be motile or non-motile, which means they can move or they can't move. So this is a picture of their shapes. Spheres, which are cocci, rods, bacilli, and then spirals. So we have, there's Staphylococcus, so that's staph infection, Streptococcus, this one. Salmonella, rods, and then these are not as common ones you would know, I don't think. Okay. Cell walls being gram-positive and gram-negative are basically just how they react and to a testing, and it depends on what they're made of. You can see that gram-positives have like a peptidoglycan wall that's fairly large and the plasma membrane, and then this one, the peptidoglycan layer is smaller and it's in between like a plasma membrane and an outer cell membrane. Okay, movement is often used by flagella. So these are flagella. They look like little, tails. They can have a single flagella, a tuft, to the flagella at both ends, flagella all over, tufts at both ends. Metabolic activity, they can either be heterotrophs or autotrophs. So heterotrophs meaning that they eat food, autotrophs meaning they make their own food. Aerobes use oxygen, anaerobes, or facultative anaerobes, which means they can survive with or without oxygen. My dog might bark because I hear the mailman. Stay tuned. Here we go. Okay, growth and reproduction. There are no male or female bacteria. They do not engage in meiosis. They are typically reproducing asexually. So these questions here will be part of like the wrap-up questions at the end of this that you'll do on Google Classroom. Okay, binary fission is a form of sexual reproduction. That is when your cell doubles in size and divides in half. So you can see right here, we have a cell that's replicated, it's elongating, and then it separates. You get 2 daughter cells. Conjugation is when you have the exchange of a portion of genetic material between bacteria. It increases genetic diversity. So it's like crossing over in sexual reproduction, except they're doing this asexually. Okay, so moving on. As you guys go, I'm assuming that you're going to need to hit pause especially some of you slower writers, so that you can get everything written down. So at this point, we are on page 2. So transformation is when bacteria takes up DNA from the environment, and then DNA can be a fragment or a plasmid. So fragments look like this, like what you imagine is a fragment. A plasmid is more circular. And so like in genetic engineering, this is like a type of transformation. All right, spore formation. Bacteria form spores when environmental conditions are unfavorable. A spore is a coat that surrounds the bacteria. It protects the bacteria from stress like heat and chemicals. All right, so big name spore formers and where you might find them. So anthrax, right here, is in wild and domestic animals. It's rare in the United States. Botulism can be found as bacteria that might be found in improperly canned foods. Tetanus can be found in the soil, also on rusty nails. Okay, moving along. Spore formers in the news. This is just kind of a separate note. So the unvaccinated boy nearly died from tetanus, and the cost of his care was almost $1 million. So this is just to show you that vaccines are important, and they help protect you from spore formers like tetanus. Okay, important functions of bacteria. Decomposition, right here, nitrogen fixation. So decomposition is when nutrients are recycled in the ecosystem. It involves digestion of dead organic matter and then the return of nutrients into the soil. Bacteria share the role with insects and fungi, like mushrooms. Okay, so stages of decomposition. You have fresh, dead pig, then bloat occurs, active decay, advanced decay, and then your dry remains. So there's a little video clip I'll post too. You guys can watch those or you don't have to. Okay, nitrogen fixation, when plants have a symbiotic relationship with nitrogen fixing bacteria. So remember that the nitrogen in the air is not usable until it's fixed. So bacteria change nitrogen gas to ammonia and other nitrogenous compounds. Plants then convert these compounds into proteins. Human uses of bacteria include mining, oil spill cleanup, biotechnology, intestinal health, and production of food. So bacteria sound like they're really bad and nasty for you, and they are, but they can also be helpful. Okay, so I think we're going to stop there for this video. Yeah, I just thought this was kind of an interesting picture. So an 8-year-old's hand after playing in the dirt. There's lots of bacteria out there. Okay, so we'll stop here, and I'm going to label this video Unit 11 bacteria, and then we'll do another video on viruses after that for part 1.